Okay. Um, I have given you guys a lecture that you were interested in on chemical signaling, on how it is that neurons, well, you wanted to know about how neurons signal, and you knew that like neurotransmitters and receptors and all these things that like you read about in, even in popular culture, you knew that those are a big deal in like kind of understanding how brain cells like work. Like they're set up in this giant communicating network. You knew that there's like some electrical parts of that, like you can record brain waves or EEGs, electroencephalograms, to record this electrical component. And yet, but there's also receptors and drugs and things. So there's like chemical components. And you guys just kind of were asking to sort of have an understanding where about like signaling and how neurons do what they do. And there's a lot of ways you can go about answering that, but a neuronal signaling is kind of like, if you're going to kind of try to address like how neurons are doing what they're doing, I think one of the best ways to go about it is to just understand like they are insane masters of signaling and communication, electrical and chemical. And all cells do that. Essentially all cells have the capability to do that. Even a liver cell does all sorts of chemical signaling. And so it's interesting that neurons aren't doing anything unique, but they are like grand masters of this, and so much so that, again, the complicated interconnections of electrical and chemical signaling and all the insane like dynamics and complexities, again, we think that somehow like all that insanity is leading to emergent properties of like consciousness and all these things that we can't even understand from the nuts and bolts and molecular pieces. We're not there yet. But one good way to just understand what we do know, and then maybe to think about how can these nuts and bolts lead to such crazy things like a thought or a mood or a memory or consciousness, it's a starting point. I divided the lecture up into two parts. There was there's going to be the electrical. That's what I have not done. Electrical signaling component, that's coming. But what I did do is I finished with you the chemical component, and I gave you a good understanding of all the chemistry and biochemistry that leads to, for example, when one brain cell is communicating to another. So I kept calling brain cell 1, and all of this in here was brain cell 2, and there was a space in between them called the synapse, and this electrical signal leads to the release of a chemical signal into that space, into that synapse. So all in here, these little triangle thingies are neurotransmitters. And you can see they're stored in these vesicles, but somehow the electrical signaling leads to the ability of those neurotransmitters to get spit out into the synapse. Then you're going to have receptors in cell number 2 that span this plasma membrane here, so that when a receptor gets bound on the outside, it can somehow transmit that fact to the inside of a cell, and now you've got an intracellular, like a notification that something important has happened. And what I did in the whole chemical signaling lecture for you guys is I kind of gave you examples and told you about what we know about how these little changes in the receptors that mute through to the inside of the cell lead to biochemical cascades, so lead to a bunch of chemistry that leads to like a significant change, that ends up leading to a big change in like the state, big change in the state of neuron number 2. And it was, and the culmination of that was a whole slew of different proteins were getting altered and changed, usually by phosphorylation, and those phosphorylations do things. So this is just a big review, and again, I got an email question list from Chloe, so before I go into the electrical, I figure we might as well like really just be super solid, and if you really want to understand this, well, let's really understand it. So before we can move on to the electrical, I'm going to finish off just like tidying up the chemical reaction part by going through the questions that Chloe had. And I'm just going to read right from her email and use that to kind of just do a big review, and because any questions she had might have been holes in the way I presented it. So here we go. So Chloe says, she gathered that an electrical impulse is sent down axon from cell 1, which pushes the neurotransmitter into the synapse. So that's this electrical activity, which we haven't talked about yet. That's all coming. But yes, there's some sort of weird electricity coming down this axon. And this axon's like a tiny little wire. Again, completely microscopic. You cannot see any of these with your eye. All of this connection, this synapse is all, you can't even see it with most synapses. You can't even see with a light microscope. You really need to get into electron microscope levels. So these are tiny structures. One big brain cell may have like tens of thousands of these connections coming in. It's crazy, but we're just focusing on one. So yes, electrical signal comes in, just like Chloe said, and it causes neurotransmitter to get pushed out. Now, it's funny, but in these lectures that I give to you guys, like I try to simplify it, and just say, hey, an electrical signal comes down here, and that's going to end up causing these neurotransmitters, which are held inside, to get spit out into the synapse. But even there, just to show you how much is known, and Chloe comments on the amount of complexity, that she's surprised at how much is actually known about these, but there really is. So I could give an entire hour lecture about exactly all the details about how the electrical signal coming in causes these vesicles to release the neurotransmitter out into the synapse. You can literally talk about all the biochemistry and the chemical mechanisms that do that link this electrical signal into the chemical signal. And again, that has to be done by having the electrical signal somehow be able to trigger these vesicles to release, to fuse with the membrane here, and then dump out the neurotransmitter out into the synapse so they can bind to the receptors. And again, the mechanisms down to the chemical mechanisms in a lot of those processes are known, and literally you can look at whole hour-long lectures about SNAPs and snare proteins and all these complicated machineries, that are involved in that. Just that process alone, it's insane, and it's really cool. But anyway, we'll try to stay focused here and just keep our eye on the prize of getting a good understanding of all this chemical signaling part. So then Chloe says that activated messenger proteins that signaled for enzymes in here. So yes, so this little trigger in a receptor ends up activating enzymes. And enzymes is just a fancy word for any protein that does a chemical reaction. Protein that can facilitate a chemical reaction. So for in that cascade, I gave you one example of one such cascade where, let's just say this is a protein that can stimulate an enzyme called adenylate cyclase to start making a molecule called cyclic AMP, and that's a reaction. It's making cyclic AMP from some other molecule. So it's doing adenylate cyclase is doing some chemistry to create this small molecule. And I drew a little picture of what would be like a protein kinase A, but this is just another enzyme. It's like an enzyme cascade. And when cyclic AMP is made, by this little football kind of thing, it sticks to the protein kinase A and causes the protein kinase A to do some reaction. It's going to do some reaction. So you get an enzyme, you get a biochemical cascade, a cascade of one chemical reaction to another chemical reaction to another chemical reaction. So you're activating, and to do that, each time you're activating a different enzyme down the chain. That was something that was a little bit not clear, and I can tell from Chloe's questions. So again, she says, the neurotransmitter causes the conformational change in the receptor. That's here, and you form the little foot down here. And then that stimulates an adenylate cyclase, she says, which creates cyclic AMP, which binds to protein kinases. So that was here. So Chloe got that right. And that creates she wrote, chemical bond. The pKa, when activated, creates a chemical reaction called phosphorylation. So Chloe's got that right. Now we're down here. So your protein kinase A is going to do phosphorylation, where they're sticking phosphate groups onto giant proteins. So all giant proteins may have a little site where they can accept these phosphate groups. And if you have an enzyme that can do that can put one of these on, that's the definition of a special kind of enzyme, and we call those kinases. So kinases are very important for cells because they're by putting these phosphate groups onto proteins, they're changing the state of the proteins. And if you can do that, if you're doing all that to like lots of proteins in here, like lots of proteins are getting phosphorylated all over the place, they are now altered in their state and in their functions and what they can do. So Chloe said they can create numerous changes in that cell. And I gave you one example, and Chloe wasn't totally confused about like what the kind of thing that a phosphorylation can do. So I gave you an example of, let's say, in this cell number 2, you can have receptors that are inside the cell. In other words, they're kind of receptors they're not really receptors because they can't do anything, because they can't sense the neurotransmitter out here in the synapse because they're inside. But if you phosphorylate, like let's say receptors, I just gave you an example of the kind of things that, you know, just a very specific example of when we say, hey, there's a change happening that's meaningful or that can lead to a new function. We can't leave that vague. I have to give you a specific example of what we mean by that so you can say, oh yeah, that makes sense. So one of the many ways that cells can like change a state, there's all sorts of ways. Almost every way you can think about it is the cell is doing actually. It's kind of, you can reverse engineer, think through like so many of these processes so that everything ends up making sense. But just to give you one concrete example, I gave you the example of receptors that are not functional because they're intracellular, they're inside the cell. But when they get phosphorylated, that causes the receptor to now get put into the plasma membrane of cell number 2. So now this thing is a valid receptor which can bind something in the synapse and do its own change and maybe activate a new set of kinases. So, you know, that means like we actually have changed the state of this cell. Maybe we've enabled this cell to detect another molecule in the synapse by doing that. Just one kind of basic example of the sort of things that neurons can do when chemical signaling is leading to a functional change. So Chloe said, you made it look like that a protein was actually turning into a receptor. That's where I probably didn't go over it well enough. But yeah, you have receptors that are intracellular, meaning they're not totally valid. They can't work yet because they can't bind the thing that they're usually listening to, because they can only do that when this part here is out in the synapse, and that can't happen until this intracellular receptor gets trafficked into the plasma membrane. Now it's got this external site that can see, it's sniffing around in the synapse to now act. So now it can act like a valid receptor. It can be bound by something in the synapse. You can get its little conformational change, and now it can start activating a kinase cascade. So yeah, so let's be clear. I gave you an example of what are called intracellular receptors. Intracellular receptors can't work because they're inside, but when you traffic them to a plasma membrane, they are now functional receptors. So I wanted to clarify that. Then Chloe says, or she asks, I gathered that a phosphate is a chemical structure with water and other molecules that make it be negative charged. So, kind of, sort of, I drew a phosphate group. I'm going to erase all this. I drew a phosphate group to be a structure that looks like this. It's a phosphorus, and it's its own molecule, and it's got a bunch of oxygens attached to it. Now, and I told you that it was negatively charged. So around these oxygens, there's negative charges. But it's not like there's no water molecules inside this. This PO4, the chemical signal for a phosphate is PO4 because it's the phosphorus with 4 surrounding oxygens, and they are all bound together. These oxygen atoms are they're negatively charged because they're sort of hogging electrons from phosphorus. Again, it doesn't make sense to get too crazy into chemistry and electronegativities and polar molecules and all the things that make molecules be charged. I just let it be that this molecule in itself, because it's its negatively charged particles are distributed not symmetrically, it ends up having like a wall of negatively charged area here. And so when proteins, if you've got a big protein now that's like all packaged up and now it gets phosphorylated, that phosphate group might really change the conformation of this protein so it looks very different, so that it can now, it can now do something. It can do something It can do something else. So that I was just saying, again, just like, trying to demystify, like, so what? You stick a tiny little few atoms onto a protein and now all of a sudden the protein's magically changed? I didn't want it to seem to you guys like there's any magic here, that like the protein, by having like a big wall of negative charges on it, that negative like electric field can literally induce changes into the neighboring parts of the protein. Proteins are giant molecules compared to these phosphates. But with this wall of negative charge, you can induce conformational changes in a protein to make them look and act very different. And again, that's all part of this thing of being like meaningful change to a cell. If this now protein can now all of a sudden do something new because of that conformational change, that phosphorylation had a meaningful effect. So Chloe was having questions about that, so I just wanted to clarify that. Does it find the proteins to bind to just because it magnetizes and moves towards it? So that's a good question. I told you that the kinases that do this phosphorylation, pop the phosphate group on. So what's interesting here is that enzymes are very specific molecules. So like if this protein is one that and again, I just used protein kinase A as an example of just one of the zillions of kinases that are in cells. I gave that as the example. But let's say protein kinase A did this phosphorylation here. The protein kinase A will actually be able to grab onto the phosphate group, and it'll be able to sort of hold on to this molecule very specifically, and it brings the phosphate group together very close to this protein of interest. So protein kinase A can do this phosphorylation because it actually has sites in its structure that sort of recognize a protein that it can phosphorylate. So it sticks to the protein. It also sticks to phosphate group. So it's bridging. It's like bringing friends together. It's bringing the phosphate group to the party of, to be near this protein, and then it actually can create the bond between them to permanently stick them together. And after that happens, this protein kinase A comes off to do another reaction, and now this protein is phosphorylated, and it's now altered by this reaction. So again, I didn't go into those details, and Chloe was asking questions. It just seemed magical that, like, she was like, how does this phosphate just like get on to the protein? But that job is actually done by the kinase, where it bridges the proteins of interest whose job it is to phosphorylate with a phosphate group. It actually sticks to them. It's all mechanical, and it does that chemical reaction by juxtaposing phosphates with proteins of interest. And proteins of interest in the literature, if you're reading about this, those are always called substrates. So the proteins are substrates for their kinases. So kinases bridge a substrate protein to a phosphate group and do a phosphorylation of their substrate proteins. All right, so that's just like a lot of really nitpicky, nitty-gritty, clarifications of some of the things that I just glossed over in that first lecture on chemical signaling. And sure enough, it tended to be the things that I glossed over that Chloe was asking very specific questions, because the things I glossed over and didn't give you molecular detail seem mysterious. It's just like, well, how did that happen? But the lectures are always going to be like that. I always have to sort of pick and choose what to gloss over and what to go into crazy details on. But in this case, I figured I would put together a whole nother lecture kind of answering those very detailed questions because you guys wanted very good understanding of this. So before I move on to electrical signaling, which I'm also going to do today, I just wanted to answer some of these mysterious, maybe holes that I glossed over in the first 2 lectures on chemical signaling. So having done that, I'm going to end here, and I'll be ready to now talk about the whole electrical signaling part and compare and contrast that to the chemical signaling, and hopefully then this whole idea of like how neurons are really working to do these 2 types of signaling and marry them together to to just do all this super complicated stuff that leads to weird emergent properties. Again, that's where we're going. We're just trying to understand the groundwork to begin to understand the crazy complexity, and we believe it is the complexity that leads to the magic of what the proteins are doing. So, of what neurons are doing, I mean. So, I will end it there.